Science at its core is a journey of discovery, a quest to understand the intricate workings of the universe around us. It is a systematic and logical approach to unraveling the mysteries that have captivated humankind for millennia. This incredible journey of scientific exploration is guided by a powerful tool, the scientific method. The scientific method is not merely a set of rules to be memorized. It is a way of thinking, a framework for investigating the world with curiosity and rigor. It empowers us to ask questions, challenge assumptions, and ultimately arrive at a deeper understanding of the natural world. Every scientific breakthrough, every technological marvel begins with the simple act observation. It is the spark that ignites the flame of curiosity, the impetus for countless scientific endeavors. Observation involves using our senses, sight, smell, touch, taste, and hearing, to gather information about the world around us. Sometimes we stumble upon something unexpected, an anomaly that piques our interest and sets in motion a chain of scientific inquiry. Other times, observation is a more deliberate process, carefully planned and executed to test specific hypotheses or gather data about a particular phenomenon. Consider the simple act of watching a sunset. A casual observer might simply appreciate the beauty of the colors painting the evening sky. A scientist, however, might be drawn to the way the sunlight refracts through the atmosphere, scattering different wavelengths of light and creating the vibrant hues we observe. Observation, while crucial, is only the first step in the scientific method. Once we have observed a phenomenon that piques our interest, the next critical step is to ask the right questions. These questions should be clear, concise, and testable, meaning that we can design experiments to find answers. The nature of the questions we ask will shape the course of our investigation and ultimately determine the knowledge we gain. For example, if we observe that plants grow taller in sunlight than in the shade, we might ask, does the amount of sunlight a plant receives affect its growth? This is a testable question that can be investigated through a well-designed experiment. However, if we were to ask a vague question like why do plants grow, we would find it much more challenging to design an experiment that could provide a definitive answer. With our questions clearly defined, we now venture into the realm of hypotheses. A hypothesis is a tentative explanation for an observation, phenomenon, or scientific problem that can be tested by further investigation. It is essentially an educated guess, a proposed solution to the question we have posed. A good hypothesis should be testable and falsifiable. Testable implies that we can design experiments to collect data that either supports or refutes our hypothesis. Falsifiable means that there must be a possible outcome of our experiment that would prove our hypothesis incorrect. This concept of falsifiability is crucial to the scientific method. If a hypothesis cannot be disproven, it lies outside the realm of science. Section 5. Predictions based on hypothesis. Once we have formulated a testable hypothesis, the next step is to make predictions about what we expect to observe if our hypothesis is correct. These predictions provide a roadmap for our investigation, guiding the design of our experiment and helping us interpret the results. Predictions are often framed as if-then statements. For example, if our hypothesis states that increasing the amount of sunlight a plant receives will increase its growth, our prediction might be: if we expose one group of plants to more sunlight than another group, then the group exposed to more sunlight will grow taller. These predictions provide a clear and measurable outcome that we can use to assess the validity of our hypothesis. Section 6. Experimental design, a controlled approach. To test our hypothesis and evaluate our predictions, we must design a well-controlled experiment. A controlled experiment is a scientific test that is directly manipulated by a scientist in order to test a single variable at a time. The variable is the factor that is changed or tested in an experiment. There are two key elements to a controlled experiment. 1. The experimental group. This is the group in which the variable being tested is changed or manipulated. 2. The control group. This group is identical to the experimental group in every way, except for the variable being tested. By comparing the results of the experimental group to the control group, we can determine if the variable we manipulated had a significant effect on the outcome. Section 7. Data analysis and interpretation. As our experiment unfolds, we diligently collect data, meticulously recording our observations and measurements. This data can take many forms, from numerical measurements to qualitative descriptions, depending on the nature of our experiment. Once we have collected a sufficient amount of data, we embark on the crucial task of data analysis. This involves organizing, summarizing, and interpreting the data to identify patterns, trends, and relationships. We might calculate averages, create graphs, or use statistical tests to determine if the differences we observe between the experimental and control groups are statistically significant. The goal is to extract meaningful insights from the data, to see beyond the raw numbers and discern the story they tell. Section 8. Drawing conclusions. Hypothesis versus theory. Armed with the insights gleaned from our data analysis, we can now draw conclusions about our hypothesis. Do our results support or refute our initial explanation? If our results are consistent with our predictions, we can say that our hypothesis is supported. However, it is crucial to remember that we cannot definitively prove a hypothesis true. Science is an iterative process, and new evidence may emerge that challenges our current understanding. If our results contradict our predictions, our hypothesis may be incorrect. This does not mean our experiment was a failure. It simply means we have learned something new and can refine or formulate a new hypothesis based on our findings. Over time, as a hypothesis withstands rigorous testing and accumulates a substantial body of supporting evidence from multiple independent researchers, it may eventually be elevated to the status of a scientific theory. Section 9. The importance of repetition. In the realm of science, repetition is paramount. A single experiment, no matter how well designed, cannot definitively prove or disprove a hypothesis. The results of a single experiment could be due to chance, experimental error, or other uncontrolled variables. Therefore, it is essential to repeat experiments multiple times, ideally by different researchers, to ensure that the results are reliable and consistent. Each replication increases our confidence in the findings and strengthens the validity of our conclusions. This emphasis on repetition underscores a fundamental principle of the scientific method skepticism. This healthy skepticism helps ensure that only the most robust and well-supported ideas stand the test of time.